Welcome back to the deep dive. It is Thursday, February 5th, 2026. And, I want to start today with a premise. It might sound a little controversial, especially with all the hype we've seen in biotech.
I'm always up for a little controversy. Go on.
I think we are finally done with the folding era. For years, the headline has just been the same thing over and over, right? AI predicts protein structure.
AlphaFold solves the shape.
Exactly. We treated it like it was the finish line. But looking at this massive stack of papers and code we have on the table today, that just feels, I don't know, insufficient.
It does.
It feels like we were all obsessing over a static photograph and then suddenly someone handed us a video camera and a whole set of sculpting tools.
That's not controversial at all, actually.
Yeah.
I think that's exactly what the data is telling us. We're seeing a really hard pivot, even just this week.
A pivot to what?
We're moving from predictive biology, just asking, what does this thing look like, to generative world models. We're not just observing the protein anymore. We're actually simulating its thermodynamics, its movement, and we're designing interactions completely from scratch.
And that's the thread, right? That's the thread running through everything today. We've got a lot to get through.
We do.
We've got Protenix V1, which is taking on the AlphaFold monopoly with a very specific kind of brute force.
Then there's OMTRA, which unifies the whole drug design process into one model.
And distributional graph former, or DIG, which is finally getting at the problem of protein movement.
Plus a quick look at something called O-design, which is kind of trying to wrap all of this up into a single world model.
Right, but let's start with the elephant in the room, or the new 800-pound gorilla, Protenix V1.
From ByteDance Seed, yeah.
And the headline claim is just so straightforward. They've matched, and in some really crucial areas, beaten AlphaFold 3.
Which is a bold claim. I mean, AlphaFold 3 has been the gold standard since DeepMind dropped it. But what makes Protenix so interesting isn't just the raw numbers.
What is it then?
It's the philosophy. It's completely open source.
Okay, but hold on. We see state-of-the-art claims every single week. We beat AlphaFold by 0.5%. And then you dig into the footnotes and, the benchmark was cherry-picked.
Right. Or they tested it on easy proteins.
So how is this different? Why should you or I trust these numbers?
You have to look at how they graded themselves. They spend a huge chunk of the paper on this thing called fold bench. Now, historically, benchmarking in this field has been, let's just call it optimistic.
Optimistic how? Like, grade inflation.
Exactly, like grade inflation. Imagine you're testing a model on a really complex protein assembly. If the model runs out of memory, an OM error just crashes.
Yeah.
Standard benchmarks would often just, exclude those samples from the final score.
Wait, so if you fail the test because the question was too hard, it just doesn't count against your GPA?
Precisely. It made inefficient models look way better than they were because they were only being graded on the easy stuff they could actually handle.
So what did Protenix do?
They introduced fold bench corrected. They forced a common intersection of samples. It means if model A crashes on a protein, but model B solves it, that protein still counts for both. It's a penalty for not being stable. It's a real apples-to-apples comparison on the hardest questions out there.
And when you strip away that grade inflation...
Protenix V1 comes out on top, specifically in the categories that matter most, like antibody-antigen interfaces and protein interactions. It's beating AlphaFold 3.
Yeah.
And it's way ahead of other open-source models like Chai1 or Boltz1.
That's impressive. But for me, reading this, the accuracy wasn't even the real aha moment.
Oh.
The thing that felt like a genuine shift was something they called inference time scaling.
Yes. This is the paradigm shift. This is the most important part of the whole paper.
So explain this, because we're all used to the old way, right? You put in a sequence, you get out a structure, you run the software once, you get an answer, you're done.
Right, those are static predictors.
Yeah.
You get the model's best guess, and that's it. Protenix is different. It acts more like a reasoning model. Think about how you can ask a large language model to think step by step.
Okay, so it's actually thinking about the structure.
In a mathematical sense, yeah. Protenix lets you increase the number of random seeds, basically different starting points for the generation. And they found this incredible log linear relationship between the compute you use and the accuracy you get back.
So you're telling me if I'm willing to pay for more GPU time, I get a better biological answer.
That's it. It turns compute directly into quality. Let's take those antibody-antigen complexes. Super important for drugs, but notoriously hard to predict.
Okay.
If you run Protenix with just one seed, one quick pass, your success rate is about 36%, which is okay, not great. But if you scale that up to 100 seeds, If you let the model really explore the problem space, the success rate jumps to nearly 47%.
That is a massive delta. In drug discovery, that's the difference between a failed project and a clinical candidate.
It completely changes the workflow. The model isn't a magic 8-ball anymore that you just shake and hope for the best.
It's a search engine.
It's a search engine. If you have a high-value cancer target, you absolutely need to hit. You don't just take the first output. You burn the GPUs. You spend the compute budget, and you essentially buy yourself a higher probability of being right.
It's like the industrialization of scientific discovery. You can trade electricity for certainty.
Great way to put it.
Yeah.
And they made it practical, too. They released 2 versions, one trained on old data from before 2021, just to be fair, in the benchmarks.
Okay.
But they also released a version trained on data right up to June 2025.
So for a chemist working today, this model actually knows about the last few years of science. It's not frozen in time.
Exactly. It's a modern tool for modern problems.
So Protenix gives us the map, a super high-fidelity structure of the target. But, knowing what the lock looks like doesn't give you the key.
Right. And that's where this next tool, OMTRA, comes in.
This one is from the University of Pittsburgh and CMU.
Right. If Protenix is the cartographer making the map, OMTRA is the architect who's designing the building that fits on that map.
The paper calls it a unified framework. Now, usually when I see unified in tech, it just means they glued 3 different tools together with a new logo.
I know that feeling. But this is actually architecturally unified.
Hmm.
The big problem in drug design right now is fragmentation. You've got one tool for docking, another for designing side chains, a third for checking physical validity. It's a mess.
And OMTRA fixes that.
It uses something called multimodal flow matching to do all of it in a single graph representation.
Okay, let's pause on flow matching.
Hmm.
I've seen this term pop up more and more, sort of replacing diffusion. What's the difference?
It's a subtle but really powerful one. Think of diffusion-like for images as taking a photo, slowly adding noise until it's static, and then teaching the AI to reverse that whole process. It works, but the path from noise back to the image can be kind of winding and inefficient. Flow matching is more direct. It learns a vector field, like a map of wind currents, that transports the noise directly to the final data. It's a straighter path.
So it's faster, more stable?
Generally, yes. And OMTRA applies this to everything at once. The protein, the drug, the ions, even modified residues, it's all one connected system. It generates the context and the solution together.
To learn that, it must have needed a ton of data. They mentioned this Pharmit dataset. I'd never heard of it.
It's staggering. They processed public chemical libraries and generated the 3D shapes for 500 million molecules.
500 million.
500 million. It's one of the largest 3D molecular data sets ever. So before OMTRA even tries to design a drug, it has this deep intuitive sense of what a real molecule is supposed to look like.
But the feature that really got me, and this goes back to our theme of control knobs, was the pharmacophore conditioning.
Yes.
Because usually generative AI is just a black box, right? You say, make me a binder, and you just hope.
Right. And chemists hate black boxes. They have intuition. They can look at a protein pocket and say, I know I need a hydrogen bond donor right here and a greasy ring over there.
So OMTRA lets you just tell the AI to do that?
It does. A pharmacophore is just that abstract map of features. You can feed those constraints directly into the generation. You can say, design a drug, but it must have these features at these exact coordinates.
That's like prompt engineering, but for chemistry.
Exactly. Instead of in the style of Van Gogh, it's with a hydrogen donor at coordinate X. And it works. When they added those constraints, interaction recovery went up by 10 percentage points.
That's huge.
And even better, it cut the amount of sampling needed by 56%.
Because you're not just guessing anymore.
You're not guessing. You're collaborating with the model. You're giving it the blueprints and letting it fill in the details.
Okay, so let's recap. We have structure and accuracy with Protenix. We have design and control with OMTRA. But both of them still have what I call the statue problem.
The statue problem.
We're treating proteins like these rigid, perfect sculptures, but biology wiggles, it vibrates, it breathes.
And that statue problem is the single biggest reason drugs fail. We design a key for a statue, but in the body, the statue moves and the key falls out.
So that's where this next one, the distributional graph former, or DIG, from Microsoft Research comes in.
It changes the entire game.
They use BRAF kinase as their main example. That's a classic cancer target, isn't it?
It is. BRAF is basically a switch that tells cells to grow. And like any switch, it has an on position and an off position. Structurally, that's an activation loop being open or closed.
And if you run BRAF through AlphaFold, what do you get?
You get a broken switch. The model predicts a structure that's just an average of the two. It's a shape that doesn't actually exist in nature. It's like a long exposure photo of a swinging door. It looks half open, but you can't use it.
And a drug designed for a half-open door won't work.
Not at all. You need to target one state or the other.
So how does DIG solve this? I mean, we've had molecular dynamics simulations for decades. Why do we need AI for this now?
We can do it with MD, but it's incredibly expensive. The reason is that MD is statistically dependent.
Break that down for us.
Okay, imagine you're trying to map a mountain range by hiking it. With traditional MD, you have to walk every single step. You calculate every femtosecond of movement. If there's a huge mountain in your way, your simulation might get stuck in one valley for millions of steps, just vibrating, never getting over the peak.
You spend years of compute time just walking in circles.
Pretty much, yeah. DIG uses statistically independent sampling. It's a generative model, so it doesn't have to walk the path. It learns the probability map of the entire landscape. It can just teleport paratroopers into all the different valleys at once based on how likely they are to exist.
Predicting the whole distribution of shapes directly.
The equilibrium distribution, exactly. And the speed difference is just, it's absurd. For the SARS-CoV-2 protease, a classic MD simulation to get the full distribution took 70 GPU years.
70
years. DIG recovered that same distribution in 18 GPU days.
That's the difference between theoretically possible and we did it this week.
And the insights you get from that are profound. The example that really hit me was about the Omicron variant of COVID.
Oh, yeah, I remember this. When Omicron came out, the static structures looked pretty similar to the original virus. Everyone was wondering, why is this thing so much more infectious?
The photos looked the same, but the movie was different.
How so?
When they ran DIG on Omicron, the distribution showed that its spike protein spent way more time in the specific open conformation that's primed for infection.
So it wasn't about the shape it could be in, it was about the probability of it being in that shape at any given moment.
Precisely. The static models missed it completely because the average shape was almost the same. But the distributional model saw immediately that the virus had evolved to be open for business more often. That fourth dimension probability, or time, is what DIG unlocks.
That's wild. You can see the ghosts of the protein, the rare states that are the key to actually drugging it.
And that connects to the last paper, O-design. We can just touch on it. But it's trying to apply this logic to the entire biological world.
The world model idea.
Right. Protenix is protein-protein. OMTRA is protein drug. O-design is trying to do all-to-all. DNA binding proteins, RNA interactions, ligands binding to DNA.
Oh.
A single generative framework for the whole interactome.
It really feels like we're watching a convergence of all these tools.
We are. I mean, think about the toolkit we just laid out. Protenix gives you the high-res map and a compute for accuracy knob.
Right.
OMTRA gives you the architect's pen, letting you put your human intent directly onto the design.
And DIG gives you the physics engine, showing you how the machine actually moves.
It moves biology from a discovery science, let's see what's under the microscope, to a true engineering discipline. You specify parameters, simulate dynamics, and generate a solution.
We're no longer just passive observers.
Exactly. And that brings me to a final, maybe provocative thought.
Let's hear it.
We talk a lot in this industry about undruggable targets. Usually that means a protein is too floppy or it doesn't have a nice clear pocket in its static structure.
Right, a smooth surface, nothing to grab onto.
What if DIG lets us see the entire equilibrium distribution? And we see that for, say, 1% of the time, a hidden cryptic pocket opens up for just a millisecond.
And OMTRA lets us design a drug that specifically binds to that transient state.
Then undruggable ceases to have any real meaning.
The definition is about to shrink dramatically.
We're moving to a world where if a protein exists and it moves, we can probably find a way to jam the gears. We just needed a camera that was fast enough to see the opening.
That is a fascinating reality to be heading into, and probably a terrifying one if you're a virus.
Let's hope so.
We will put links to Protenix V1, OMTRA, DIG, and that massive Pharmit dataset in the show notes for anyone who wants to download a few terabytes of molecular data.
It's definitely worth exploring if you've got the drive space.
Thanks for diving in with us. We'll see you next time.